So, first of all, let me introduce Sharon Willis to you all. Sharon is the Director of Production at Integral Molecular, and she leads the Lipoparticle Production team. And she has studied membrane envelope viruses extensively over the past few years. So Sharon has been working with biosensors for over 12 years, and she is an expert in the use of lipoparticles with biosensors. So Sharon has earned her BA from Gettysburg College, cum laude, and she earned her PhD in chemistry from the University of North Carolina at Chapel Hill. And Sharon was actually involved and is a co-founder of Integral Molecular. She's also worked as a research specialist at the University of Pennsylvania, where she purified and crystallized and characterized the herpes simplex virus envelope protein, glycoprotein GD, which was a culmination of 20 years' work. So it's with a great plot of our Become an SPR Expert webinar series. She's actually the last presenter for 2009. So Sharon, with that introduction, I will pass over to you. Thank you very much, everyone.
Okay, thank you, Laura, and I'd like to thank Bio-Rad for inviting me to give this webinar today. So today I'm going to talk about novel technologies for studying membrane-protein interactions. Membrane proteins are important because they mediate cellular communication, and from a commercial standpoint, they're important because 30 to 40% of the drugs on the market today target membrane proteins. However, membrane proteins are often difficult to work with, primarily because their structure is dependent on the lipid bilayer. And trying to study them outside of the context of the lipid bilayer is a tricky and often empirical process. At Integral Molecular, our focus is on membrane proteins and developing technologies to simplify studying membrane proteins. And today, I'm going to talk about two technologies. The first involves the use of biosensors to study membrane proteins. And for this, we use one of our technologies, which is the Lipoparticle. And Lipoparticles are virus-like particles that we can engineer to contain a membrane protein of interest. I'm going to talk about two assays using lipoparticles on the biosensor. The first assay can be used to actually measure the binding kinetics of an antibody-receptor interaction. And then the second assay is more of a rapid screening of a panel of antibodies. The second technology I'm going to talk about involves mapping studies using shotgun mutagenesis mapping. This is a mapping technology that can be used to map antibody epitopes and other protein-protein interactions. So what are lipoparticles? Lipoparticles are pseudotyped, non-infectious virus-like particles that are a concentrated source of membrane proteins. On the left is a cartoon of a lipoparticle showing the membrane protein of interest embedded in a lipid bilayer that surrounds a protein core. Lipoparticles are approximately 150 nanometers in diameter and are thus perfectly suited for microfluidic applications. On the right is an electron micrograph of a lipoparticle, and the red dot represents the approximate size of a GPCR in the lipid bilayer. Membrane proteins and lipoparticles are structurally intact and correctly oriented. So how are lipoparticles made? They're produced by co-transfecting the DNA for the viral core and the membrane protein into cells. The protein is overexpressed on the surface of the cell, and the viral core assembles within the cell and then buds through the cellular membrane, pinching off a piece of that membrane that contains the membrane protein of interest. The lipoparticles are then released into the culture supernatant. And below is an EM depicting this budding process. Lipoparticles are purified from the culture supernatant and resuspended in buffer. A solution of lipoparticles will have an opaque appearance because the particles are particulate, but they can be treated as soluble proteins. 1 microliter of lipoparticle can contain as many specific membrane proteins as 10,000 cells expressing 1 million receptors each. This slide shows a comparison between lipoparticles and membrane preparations. The top are dynamic light scattering profiles of each. On the left is the dynamic light scattering profile of a purified batch of lipoparticles. You can see there's one homogeneous peak centered at about 150 nanometers in diameter. On the right is a dynamic light scattering profile of membrane preps, And you can see that membrane preps tend to be more heterogeneous in size and distribution. On the bottom are EMs that support the DLS data. You can see that the membrane preps are more heterogeneous than the lipoparticles. Another advantage that lipoparticles have over membrane preps is that all of the receptor proteins are correctly oriented, which may not be the case for membrane preps. In addition, lipoparticles are free of any cytoplasmic contaminants. Radioligand binding assays show that the receptors are conformationally intact in the lipoparticles. They bind their native ligands with the same affinities as published for cell-based assays. And these radioligand binding assays also show that the receptors in the lipoparticles are present at high concentrations. We typically incorporate receptors to concentrations of 50 to 200 picomoles per milligram in the lipoparticles. Now that I've explained what lipoparticles are, I'm going to talk about their use in measuring antibody binding kinetics using biosensors. SPR-based biosensors have many advantages. They're very sensitive, and because the data is collected in real time, you can determine the kinetics of protein-protein interactions, specifically on and off rates and affinity constants. They've been of limited use, however, with membrane proteins because cells are too large for the microfluidics, and reconstitution of solubilized membrane proteins on the chip surface is a labor-intensive and empirical process. As I mentioned before, because of their small size, lipoparticles are ideally suited for the microfluidic applications, such as SPR biosensors. Most of the data I'm going to show today was collected on a ProteOn biosensor from Bio-Rad. The ProteOn has 6 flow channels that can be accessed in either the vertical or horizontal direction. This results in 36 interaction spots that can be studied simultaneously. This is a relatively high throughput biosensor. The first assay I'm going to describe is the antibody binding assay. In this assay, lipoparticles are mobilized onto the sensor chip surface, and antibody is flowed across in different concentrations so that the detailed kinetics of the antibody binding can be measured. So first I'm going to go into a little more detail about the lipoparticle immobilization. It's the first step for this assay. Lipoparticles are compatible with all standard immobilization methods, such as direct immobilization via amine coupling or capture. Capturing lipoparticles onto the chip surface is our preferred method. We do this one of 2 ways. We either immobilize a lectin onto the surface of the chip and capture the lipoparticles via surface carbohydrates, or we immobilize a capture antibody that's specific for one of the proteins on the surface of the lipoparticle. The buffer that we use is PBS with a little bit of BSA. And what's important to know is that lipoparticles are not compatible with detergent, so there's no detergent in any of our buffers. So the basic capture method is to first immobilize the capture molecule, either the lectin, which we typically use wheat germ agglutinin, or the capture antibody. Wash the surface with buffer, capture the lipoparticles, wash again, and you're ready for your antibody injections. So here's another visual of the capture process. On the ProteOn, capture is done in the vertical direction. So first, the capture molecule is covalently attached to the chip surface, and then lipoparticles are flowed across in the same direction. So in this experiment, or in this assay layout, what I'm showing is that we're going to capture, CXCR4 containing lipoparticles on the first flow channel, and then control lipoparticles on the second channel. We call our control lipoparticles nulls, and what null means is that these are receptor-free lipoparticles that are made the same way the receptor-bearing lipoparticles are, except we don't transfect in the DNA for the receptor. So these are receptor-free lipoparticles, and these are really the best thing to use as a reference in the experiments. This is a screenshot of the lipoparticles being captured onto the chip surface. We typically do these as 2 injections, and that's so that we can really maximize the amount of lipoparticles that are being captured onto the chip surface. So what's shown here is this is the start and stop of the first capture injection, and then buffers float across the surface, and then here's the start and stop of the second capture injection. We typically capture 4,000 to 6,000 RUs of lipoparticles onto the chip surface. This is actually an image of the chip surface that was taken under a microscope, and on this chip, we immobilized either green fluorescent lipoparticles or red fluorescent lipoparticles. And you can actually see the individual interaction spots on the surface of the chip in this image. So the next step is to look at the antibody binding to the immobilized lipoparticles. And on the ProteOn, the antibodies are flowed in the horizontal direction. And on the ProteOn, this can also be done in one cycle as one-shot kinetics. So 5 different concentrations of antibody, so your dilution series of antibody, plus just plain buffer, are flowed across the chip surface simultaneously. And this is a screenshot of the data that results from these injections. So on the left is a control surface. So on this surface are null lipoparticles, and on the right are sensorgrams from the flow channels containing CXCR4 lipoparticles. On the top, this shows the control negative antibody flowing over both the control surface and the CXCR4 surface. You can see there's just about no nonspecific binding. You just see the standard changes in refractive index. And then on the bottom, this is the CXCR4 specific antibody being flowed across either the control surface or the CXCR4 surface. And again, there's little to no nonspecific binding, but there is definitely specific binding of the anti-CXCR4 antibody binding to the CXCR4 surface. Then these control surfaces can be used for background subtraction, and what results is process sensorgrams, shown here. So this is a slide showing the process data, so it's been background subtracted for 2 different CXCR4 antibodies. 12G5 is a commercially available antibody, and INT11 is an in-house antibody that we co-developed using our lipoparticle technology. The data for each antibody was globally fit with a bivalent model using the ProteOn software. The black dots represent the raw data, and the red lines are the fit. And you can see they overlay each other very nicely, showing that this is a good fit of the data. And the rate constants and affinities are listed at the bottom of the slide for these 2 antibodies. Data for multiple antibodies can be graphed on an isoaffinity plot. On the y-axis are the on rate, and the x-axis is off rate. And each individual point on this graph represents a kinetics for one monoclonal antibody. Antibodies that fall on the same diagonal line have similar KDs, but this clearly shows that even if 2 antibodies have the same KD, they can have very different rate constants, binding kinetics, different on rates, different off rates. The second assay I'm going to describe is the lipoparticle binding assay. In this assay, antibody is either directly attached or captured onto the chip surface, and then lipoparticles are flowed across. This is a method that can be used to rapidly screen a panel of antibodies or hybridomer supernatants and get a relative ranking of their affinities. I'm going to show you 2 examples of this assay. In the first one, we use lipoparticles containing the GPCR CCR5 with or without an N-terminal HA tag. We captured 4 different CCR5 specific antibodies to the chip surface. 2 of them, 2D7 and 523, recognized conformationally complex epitopes on the surface of the protein. And the other 2, CTC8 and CTC5, recognized N-terminal epitopes on the surface of the protein. Then we flowed lipoparticles across this antibody surface, either our control negative lipoparticles, our null lipoparticles, or lipoparticles containing either HACCR5 or wild-type CCR5. And as you can see, wild-type CCR5 binds to all 4 antibodies, whereas the HACCR5 binds to 3 of the 4. It does not bind to CTC8 because the epitope for CTC8 is in the N-terminus of the protein, and the HA tag blocks that epitope. So this is a very sensitive assay, and it's sensitive because the binding signal is enhanced by the lipoparticle's large size and intrinsic multivalency. So you can see we overall get a very high binding response. In this experiment, we screened a panel of antibodies against CCR5, and the panel was evaluated for specificity and relative affinity. The panel includes well-characterized and neutralizing antibodies, conformational sensitive antibodies, antibodies with diverse epitopes, and a negative control antibody at the top here. So the antibodies were captured onto a chip surface. The chip surface contained a secondary antibody, so we could capture the antibodies onto the chip surface, and they were all captured to similar levels. Then lipoparticles containing CCR5 were flowed across the surface, and the antibodies were ranked by the strength of lipoparticle binding. The rankings are shown here in the bar graph, with the highest binder at the bottom, so this 2D7 down to most lipoparticles, and decreases from there. And these rankings agree with published affinities and affinities that we've measured in our lab. So this approach can be used for screening hybridoma supernatans, or really any molecule that can be attached to the chip surface. And as I said before, it's a very sensitive assay because of the size and the multivalency of the lipoparticles. Every biosensor experiment ends with regeneration of the chip because ultimately you want to be able to use your chip surface more than once. So we do regenerate these chips, and when we do these experiments in the capture format, we regenerate down to the original capture surface. So we either regenerate down to the lectin, the wheat germ agglutinin, or we regenerate down to the capture antibody, just depending on what we have immobilized on the flow channels. Regeneration is accomplished with short pulses of detergent followed by a low pH buffer. And then if it's a lectin surface, we also have to run a sugar solution across the surface to disrupt that interaction. And then the chip is ready to recapture lipoparticles. So the chip can be used multiple times for repeats of the same lipoparticle, or it can be used for lipoparticles containing different receptors. The chip surface can be regenerated at least 5 times. That's what we've determined at this point for these experiments. So next, I'm going to talk about another interaction technology that we've developed at Integral called shotgun mutagenesis mapping. This is a high-throughput mutagenesis strategy for identifying critical residues involved in different protein interactions. There are 3 steps to shotgun mutagenesis mapping. The first step is to create the mutation library. Every amino acid in the protein of interest is mutated, and each clone in the library contains a unique amino acid mutation. The library is then arrayed and expressed in 384 well plates, which can then be assayed for different protein interactions. And that's the second step of the process, is to assay for function. We can assay for functions such as ligand interactions, functional interactions, or antibody interactions. Critical residues are identified for each of the functions, and then in the third of the protein. We use shotgun mutagenesis mapping to identify the amino acids and the epitopes for the panel of CCR5 antibodies that I talked about previously. and 2 are shown here, 2D7 and 45523. Through this process, we identified 2 critical residues in the epitopes for 2D7 on CCR5 and 4 critical residues in the epitope of 45523 binding to CCR5. And these are all in extracellular loops 2 and 3. One of the critical residues for both antibodies was E172. To further analyze the importance of this residue in the antibody CCR5 interaction, E172 was mutated to nearly all of the other possible amino acids. So CCR5 containing the different E172 point mutations was tested for binding to both 2D7 and 45523. So on the graph here, this is the wild type, a stop codon, and then the different amino acid changes, all at position 172. As you can see, none of the mutations permitted binding to 45523, which is shown with the blue bars. And only the glutamine mutation permitted binding to 2D7. The 2D7 binding is shown in the red bars, and here's that change to glutamine. Now, glutamine and glutamate have very similar side chains. They only differ right here, where glutamate has a carboxylate and glutamine has an amide. The data suggests that 2D7 and 45523 interact with different aspects of the glutamate side chain. Specifically, it appears that the charge on the carboxylate is necessary for interaction with 45523, but not necessary for interaction with antibody 2D7. We then wanted to further study this interaction by looking at the binding kinetics of these 2 monoclonal antibodies to the E172 variants on the biosensor. So to do this, we made lipoparticles containing either wild-type CCR5 or 2 different variants of CCR5, either CCR5 with a mutation E172Q or the mutation E172D. So we made and purified these lipoparticles, attached them to the sensor chip surface, and then looked at the binding kinetics of 45523 and 2D7 to these different variants of CCR5. So on the left is the binding of 45523 to wild type and the E172 variants, and on the right is the binding of 2D7 to wild type and the E172 variants. And as you can see, the 2 antibodies have drastically different binding kinetics with the E172Q variant. And they both almost don't bind at all, except here at very high concentration to the E172D variant. So this data supports the shotgun mutagenesis mapping data that suggests different modes of interaction for these 2 antibodies with CCR5. So in conclusion, today I've presented 2 novel technologies for studying membrane-protein interactions. The first involves lipoparticles, primarily in biosensor applications. I described 2 different assays. The first was a rapid screening for antibody specificity and affinity, and this can be used to rapidly screen a panel of antibodies or hybridoma supernatants and get a relative ranking of their affinities. And then, following up on that, you could also do a detailed kinetic study on any antibodies of interest. The second technology, the shotgun mutagenesis mapping, this involves a comprehensive library of mutations that can be used to rapidly screen a panel, that can be used to identify critical residues in protein-protein interactions, such as antibody binding, functional assays, or other protein-protein interactions. And identified residues can then be mapped onto the structure of the protein. I want to end today by directing you to our website. On our website, under the Applications tab, you can find and download different application notes for the applications I've talked about today, and publications that we have. In addition, you can find our contact information and information about sales and service on our website. So I'd like to thank Laura and Bio-Rad for inviting me to give this webinar, and I'm happy to answer any questions.
Okay, thank you very, very much, Sharon. That was wonderful. I will like to open up the floor to any questions that we have. I'm just going to take everybody off of mute. So everybody, you're off the mute function now. So does anyone have a question that they would like to ask to Sharon? Okay,
hello.
I have a question. Hi. Go ahead.
Excellent. Nick Decker here from AstraZeneca. I have a question to Sharon on the lipoparticles. Could you comment on the high density of receptors that you mentioned there? Typically, receptor densities of 50 to 200 picomoles per milligram in your lipoparticles. Is there any way that you have enriched the receptors in the virus? What I understand from the budding process is that the virus has the identical composition of the membrane as the regular plasma membrane. So could you comment on the high levels of receptor? Is that maybe due to the fact that the lipoparticles themselves have very low protein content?
Right. That's exactly what it is. We overexpress the receptors on the surface of the cell, so we really push the cells to maximize that expression. And then the lipoparticles themselves just have a much lower protein content than the cells. They don't have all those extracellular proteins contaminating the prep.
Okay, thank you very much.
We've had a question on the text here. What is the size of the lipoparticle?
Lipoparticles, they're 150 to 200 nanometers in diameter. Thank you.
Any other questions there? Yes.
So with the lipoparticles being that large and the SPR wave that comes off. Can you hear me?
Yes.
Okay. And the SPR wave kind of loses its sensitivity around that range. Do you get any sort of artifacts from that? So if you have an antibody that binds to the top of the lipoparticle versus the side of the lipoparticle, would you get a different RU response?
No, because it's averaged out over the surface of the chip surface, so we don't see any artifacts. I mean, the lipoparticles, with their 150 nanometer diameter, are kind of pushing the envelope of sensitivity for SPR, but they work really well in the antibody binding.
So what is the lowest molecular weight ligand you've looked at in terms of binding to the receptor expressed in lipoparticles?
Right. A majority of our studies have focused on antibody binding. We've briefly tried smaller molecules, but again, I think because we're really close to that limit of detection, it's just not sensitive enough to see small molecules. But if you turn it around, if you could somehow attach your small molecule to the surface of the chip and then flow the lipoparticles across, that's an extremely sensitive assay. So you could screen a small molecule library that way.
What about just smaller proteins, like 20 kilodalton proteins? Would you expect to be able to see those?
Yes. Yes, I think with a high surface density of lipoparticles, that have a high concentration of the receptor protein of interest, I think we could see interactions in that size range.
Can I ask a question which is related to this?
Yes.
Do you see the binding of chemokines to the chemokine receptor?
No.
No.
We've tried. I say no, but we haven't put too much of a concerted effort onto that.
Okay, so you tried briefly?
Right now. Right. We tried briefly and it didn't work well. But since that was years ago, and it's something we need to come back and revisit.
Okay. Yeah.
Sharon?
Yes.
This is Eileen Lacey from Centocore.
Oh, hi, Eileen.
Hi, how you doing?
Good.
That data that you presented and you showed that you have obtained the similar values that what have been reported in the literature, did you publish that?
Yes. Was that for the rapid screening?
I think it was before the kinetic.
The TruD7 kinetics? I mean, sorry, the 12G5? Sorry.
Yeah, let's see, go back and...
Let me show you. This we have published.
Before that.
This is not published.
Something before that you mentioned that you say you got the same numbers of the experiments done in cells.
This one.
This one. Did you publish that?
Radioligand Binding. This is not published. It's in one of our application notes that you can download from our website.
Okay, can you send me an email and direct me to which one is that one? Thank you. Okay, thanks.
Hi, I've got a question. You mentioned that you could directly couple the nanoparticles, but you didn't then show any. What kind of RU levels do you get with amine coupling?
Right. With amine coupling, we can get similar RU levels. 3,000 to 4,000 RUs immobilized typically. What you have to really be careful with the amine coupling is the regeneration. If you bind an antibody or some other type of protein that binds very tightly, it's going to be very hard to regenerate that surface without disrupting the lipoparticles.
Okay.
Yeah.
Thank you.
You're welcome.
Any other questions?
Do you ever, with this direct binding via amine coupling on your chip surface, do you ever find that you also have the amine coupling within your receptor so that you also make bonds in there which make the receptor less functional?
No, I haven't noticed any diminished use of the surface because of that, because you're only coupling the one small part of the lipoparticle, so the whole rest of the surface is available for binding.
Okay. Yeah, thank you.
So unfortunately, I logged in late and didn't see the early slides where you're making the lipoparticles. If you still have time, could you go back and quickly review those slides?
Sure. So I'll review. Can you see the slide?
Yes.
Okay. So lipoparticles are produced in cells. So we co-transfect in the DNA for the virion core and the membrane protein of interest. So that the membrane protein of interest is overexpressed on the surface of the cell. Then the core assemble buds through the cellular membrane, pinching off a piece of that membrane that contains the protein of interest. And the particles are released into the culture supernatant, and that's where they're purified from.
Any other questions?
Actually, yes, one more.
Oh, one more question there?
When you are binding to captured antibody or your lectins, approximately how much lipoparticle do you need to inject to get the kind of RUs that you're immobilizing or binding?
You want like volume of lipoparticles that are needed?
Not volume, but amount of lipoparticle that you need to inject. Are you having to inject quite a lot of lipoparticle in an amount of binding, or in a typical preparation do you Do you use a lot of it or not much?
It's a moderate amount. The lipoparticles are large, so the process of them, it's a very slow process for them binding to the surface. So it's not a small volume, but it's not a huge volume. I mean, it's 50 to 100 microliters of a diluted solution of lipoparticles.
And how would that be diluted?
We typically, from the stock concentration, we typically dilute them, 10 to 20 folds.
And how much would you get in a stock solution?
In a stock, well, they're sold in 100 microliter aliquots.
Okay, thank you.
You're welcome.
Maybe if I can also ask a last question. If you have the surface with an antibody, for example with a 12T5 antibody, and you flush over your null lipoparticles, I tried that a few times, and then I also see a big increase in RUs of my surface. Do you have any experience with that?
No, we usually see almost no boundary. We don't usually see that.
Okay.
It's pretty much if you have BSA in your running buffer, you really shouldn't be seeing any non-specific binding.
Okay, so the amount of BSA, and you said you use a little bit of BSA. How much is that approximately per liter of buffer?
1 mg per mil is typical, but you can use higher.
Okay, 1 mg per mil, and you can use higher.
Right.
Okay, thanks.
You're welcome.
Sharon? Hello?
Okay. Hello? Go ahead.
Sharon? That was 1 mg per mil BSA?
1 mg per mil BSA is what we typically use in our running buffer.
Okay, thanks.
You're welcome.
Okay, so I think that's it for questions. As Sharon explained, you can download further information about lipoparticles and shotgun mutagenesis from Integral Molecular's website. So with that, I would like to close the webinar and thank everyone very much for their kind attention and their excellent questions. And again, thank you very much, Sharon. The webinar, I will send out the recording. So thank you.
Thank you, everybody.
Okay, thank you very much.
Bye-bye.